Welcome to Chempoetry. Today we will learn an important analytical technique called mass spectroscopy. Mass spectrometry is one of the most accurate techniques used to determine the molecular mass of a compound. It not only tells us the molecular weight but also helps us to identify unknown compounds and understand their structure. In this video, we will discuss the principal instrumentation, working process, and application of mass spectrometry in a simple and easy to understand way. So, let's begin. What is mass spectrometry? Mass spectrometry is an analytical technique used to determine the molecular mass of a chemical compound. The basic idea is very simple. First, the molecule are converted into ions, then these ions are separated according to their mass to charge ratio, commonly written as m over z or m by z. Using this information, scientists can identify unknown compounds and obtain structural information about molecules. Mass spectrometry has many important uses, like it is used for molecular weight determination, structural elucidation, compound identification, and quantitative analysis. Because of its high sensitivity and accuracy, it is widely used in chemistry, pharmaceuticals, biochemistry, environmental science, and forensic investigation. Example: molecular weight calculation. Let's understand mass spectrometry with a simple example. Consider Neopentane, whose molecular formula is C5H12. To calculate its molecular weight, carbon has an atomic mass of 12 and the hydrogen has an atomic mass of 1. We know it very well. Therefore, 5 multiplied by 12 equal to 60, 12 multiplied by 1 equals to 12. Adding them together gives the 72. So the molecular weight of neopentane is 72. In mass spectrometry, most molecular ions carry a single positive charge, which means Z equals to 1. Therefore, m by Z equals to 72 divided by 1, which is 72. Hence, the molecular ion peak of neopentane appears at m by Z ratio equal to 72. This peak helps us determine the molecular mass of the compound. Now, let's understand the basic principle. In mass spectrometry, a beam of high energy electron strikes the simple molecules. These energetic electrons remove one electron from the molecule. As a result, positive charge ions are formed. Some of these ions remains intact while others break into smaller fragments. The overall process can be summarized as sample enters the instrument, then ionization occurs, the ions are accelerated, next they are separated according to their m by z value. Finally, they are detected. A simple mnemonic to remember this sequence is IASD. I stands for ionize, A stands for accelerate, S stands for separate, D stands for detect. Remembering IASD makes the entire process easy to recall during the exam. Let's see how the molecular ion is formed. The reaction can be written as M plus electron gives M positive radical plus 2 electrons. Here, M represents the original molecule. M positive radical written as M plus dot. It called the molecular ion or parent ion. This molecular ion is very important because it retains the same molecular mass as the original molecules. It possesses a positive charge and also contains an unpaired electron, which makes it a radical cation. The molecular ion peak usually provides information about the molecular weight of the compound. After formation, the molecular ion is often unstable. To achieve stability, it breaks into smaller pieces. This process is known as fragmentation. The molecular ion can produce a positively charged fragment ion and a neutral radical. For example, M positive radical can break into M1 positive and M2 radical, or it can break into M2 positive and M1 radical. Only the charged fragments are detected by the instrument. Neutral radicals are not detected. Different compounds produce different fragmentation patterns, which become very useful for identification. Now the question is, why is fragmentation so important? Fragmentation produces characteristic peaks in the mass spectrum. These peaks act like a molecular fingerprint. Just every person has a unique fingerprint. Every compound has a unique fragmentation pattern. By studying these peaks, scientists can identify the structure of unknown molecules. There are three important types of peaks that we should remember. First is the molecular ion peak or M positive peak, which gives molecular mass information. Second is the base peak, which is the tallest and most intense peak in the spectrum. Its intensity is considered 100%. Third are the fragment peak, which arises due to the breakdown of the molecular ion and provide structural information. Together, these peaks helps us determine both the identity and structure of a compound. That's all for today's video. In this lecture, we learned the definition, principle, molecular ion formation, fragmentation process, and importance of mass spectrometry. If you find this explanation helpful, please like the video, share it with your friends, and subscribe to Chempoetry for chemistry concept explained in the simple language. Thank you for watching. Stay tuned and keep learning.